All right. So, couple of quick things. One, as everybody knows, exam Friday. It's mostly written. I did use about half of the questions are ones that are coming from you guys or some form of those. So the extra credit will come after after you take the exam on Friday. So that's one. Take home is posted. Due next week on Friday. That's two. And then next week we'll be working on blogs all week. And I do want to take maybe like 5-10 minutes today to at least introduce that to you so that you can at least start thinking about ideas. Not necessarily working on it, just thinking about ideas for it. The last thing that I want to make sure to mention is that, and I've seen some people have already done this, but I always offer extra credit to the class if a certain number of people will do the end of the semester evals. This is not a way for me to get favorable ratings. This is just a way for me to get the comments in general so that I can figure out next time we run this course, what do I need to do to improve it? Or what do I need to do in general to improve? So take the time to do that. I think they're due next week on Sunday night, so you've got another 10 days, and I'll make sure to remind again of it down the road. Where do we want to begin today? Should we start with 5 minutes of just going over the blog briefly, and then we'll spend the rest of the time doing other stuff? I didn't want to do that. Okay. So I posted this last night. It may be a little rough because I wrote it yesterday as well. So if there are questions about it, please ask, because I'm sure there are going to be mistakes in it. But the idea of this is to write something that is going to be presented to a general audience, and of course that form of presentation is writing. So we're going to use a website that I've used in the past for this because I'd like to make these public rather than private entities once they're done so that they can live on. My plan is for you to have a draft completed by the 10th, which is next week, Thursday. We can talk about specific times later. On our last class, we'll peer review, so I'd like to have everybody give feedback by that next class so that we can spend time discussing, and then the final draft is due that Wednesday at finals. Okay? And that is technically like your final project for the semester. As a benefit for us having this all next week, there's coffee every day next week. So, all right.
So Tuesday and Thursday too?
Sure. Come on to my office and I'll have a pot brewing. So the idea here is that by this point of the semester, you have spent a lot of time studying just the generals of virology and some more specific features. In the past, when I've had students do this particular assignment, what I've asked them to do is to describe the life cycle of one virus. And it was more of a creative writing exercise. So if you look at some of the examples, there are different ones where, like, one is written like a love letter. One is written as if it's a newspaper feed about a virus hijacking a mosquito and using the mosquito as a way to infect a human. What I'm going to do is I'm going to open this up a little bit because I don't necessarily feel like focusing just on life cycles is the right thing to do here. So what I want to do is give you the opportunity to pick something that you and a partner, and this does not have to be the same person you worked with on the poster, but something you and your partner want to look at further. So it could be, for example, it could be the life cycle of a virus we haven't talked about, or it might be looking at, for example, new drugs that are being used to treat Ebola infection. Okay? It should be informative. But at the same time, with something like this where you're trying to write to a general audience, it also should be written in a way that is catchy And what I mean by that is you want to hold the audience's attention as long as possible. So the rubric, I think, tries to reflect that, and that things that I consider to be bad would be like writing it like a textbook. It's boring, right? So try to avoid that. I think we can pitch this towards 100 level students. So assume that they have some basic understanding of cell biology, some basic understanding of genetics, so that you don't have to go through and define every little bit of nuanced piece. But what I am looking for you to be able to do is to describe, use analogies or examples as a way to describe some of these more complex topics of virology. And I think that's a really good measure of how well do you actually understand this information. Okay? I'm not putting a cap on how many references you need to have, either a minimum or a maximum. I'm just going to expect that you guys are referencing things that you're using for this. You're welcome to use primary literature, review articles, other reliable sources as a way to help you guide this assignment. Let me see if there's anything else I want to point out. Everybody should have gotten the link to the WordPress site last night, so you'll just have to set up an account when you go on there for the first time, and then I can start to show groups or people individually how to create these blogs, but it's pretty darn easy. If you want to work on it collaboratively, what I would suggest is set up a Google Sheet, or sorry, a Google document, or use Word Online as a way to share the file back and forth, and then just copy and paste it all into the blog when you're ready to start doing some formatting. Images are incredibly helpful here. The WordPress website makes it very easy to not only upload images or copy a URL to upload the image, but it also allows you to put captions in pretty darn easily without having to fuss with formatting. And so that's a couple of features that I would think that folks are going to use. And then for the peer reviewing, we're going to do this in a couple of ways. So one, I'm going to expect that every group peer reviews another group's blog. I've asked Paige, and I haven't heard from Bill or Cammie yet if they would be willing to peer review too. I'm not going to ask you in public, but Paige has said that she'd be willing to peer review as well. And so she's going to give some feedback to some groups to get a second set of eyes on all of these. Okay? And then finally, I will grade these, and then, like I said, I'm going to have a couple of former students of mine help to grade them as well. All right, so that we can get a couple sets of eyes on these. The instructions here are in somewhat a little scattered. Admittedly, they might be a little vague. This may seem very open-ended, and that's because I want you to take ownership of it. I don't want to prescribe what you have to do. Really, all I've got is just the rubric down here, and the rubric is kind of a modified version of the one that we used for the posters. So you'll see that the sections that I'm looking for on this one are tone, basically, is it written in a narrative style or not? Does it carry the audience's attention throughout? The descriptions are kind of the big piece. This is really where I'm assessing, do you understand the material that you're talking about? And then visuals, lit cited, and then the last one is organization and grammar. So there are some similarities to how the poster is. I've modified this, so take a look at it over the next few days. And then on the following page, you'll see how this correlates to a letter grade. It's a 50-point assignment. Okay, so I don't anticipate there are going to be many issues with this, but like I said, as we get started next week, feel free to begin to ask questions as we get going with it. All right. Any questions right now, though, about what the expectations are? Shania?
So are we supposed to still write it as a creative writing effort, That's a good question. I haven't put it specifically as that, but I think that what I'm asking you to do is to hold the audience's attention. So if that is in the format of a creative writing assignment, go for it. Go for it. If you think you can achieve it in that way, that's perfectly fine. What I'm asking you to be able to do is try and balance some of that creativeness and using analogies or descriptions or analogies and examples along with having some of that showing also an understanding of the material at the same time, if that makes sense. So it's a different type of assignment than what I've asked you to do in the past, where I've asked you to be expert level throughout. Now I'm asking you to take that expert level and drop it down about 3 notches and be able to explain it to Francie's class or something like that. Okay? Not genetics. Not genetics. So if that's in the form of examples, analogies to help guide that, or creative writing itself, that's perfectly fine. I have no problem with that whatsoever.
Can we work individually, or does it have to be?
That's a good question. I'll tell you what, if you want to work individually on it, I would allow you to do so. If that's what you would prefer, I have no problem with that. She asked, could you work individually? And if that's not something that I had considered, but if you want to, I think I have no problem with that. Right. So I suppose with that in mind then, if anybody would like to work with a partner but doesn't have one by, say, Saturday, Sunday, let me know and I'll try to work things out. All right. Anything else right now? Okay, let's talk HIV or Ebola or both. I'm going to just kind of let you guys run the show, at least to start, and I want to see, just in general, are there any questions that we should address first on material?
Did we ever go over anything like translation-wise for Ebola? Or I guess like we did like alternate splicing.
Yeah, so Ebola, its main mechanism for transcription again is it's going to produce those monocistronic mRNAs. We didn't really talk about translation so much. We said that because it looks like the host mRNA, it's probably going to follow a traditional cap dependent. We did say that there was an example of ribosome slippage though, which we see in HIV as well, and that was the soluble GP and the full-length GP. Those get produced as a result of ribosomal slippage. But beyond that, we just talked about it as being similar to eukaryotic mRNAs.
Did you say SGP is created from ribosomal slippage?
It's actually the full-length. I think the soluble form is created at... Yeah, I think the full length, or I think the soluble form is created about, I think the ratio is like 3 to 1. 3 solubles for every 1 full length. So that ratio is a little bit different than what we see for HIV's GAG and POL products, but the mechanism, the general mechanism is the same. And real quick, as a follow-up question then, what features do genomes have that allow for ribosome slippage? A run of use, or I think more broadly you might even be able to say a run of the same nucleotide. And then what's the other feature? Hairpin, stem loop, yes, exactly. Some sort of RNA base pairing to itself to create that structure. Absolutely. Sabrina, you had a question.
So the soluble GP that is in the lunar space, so is that released like out into the cytoplasm?
So it would actually, since it's soluble, it's just the head group of the GP. That actually would be not only... so it would be made on rough endoplasmic reticulum. And that means then it's going to go through a exocytosis process to make its way out of the cell. And instead of being embedded in the membrane and staying on the membrane, once that exocytic vesicle moves out, that's going to release it actually outside into the extracellular material. So plasma, tissue, outside into the tissue, interstitial fluid, etc.
And that's for neutralization.
So the purpose of that, again, the purpose of that soluble GP is it serves as a decoy. So the idea is that the immune system, particularly B cells, are going to produce antibodies, and that soluble GP, I'm going to pull up these slides so that we can look at this too. That soluble GP would serve as a decoy, bind to the antibodies, and therefore reduce the amount of available antibody that's able to bind to a infectious virion itself. And those antibodies we could consider to be neutralizing antibodies, as they would prevent the virus from binding to the host cell. Sorry, I'm going to move ahead real quick. There we go. So that secretion of this soluble form here would bind to antibodies and prevent the antibodies, or reduce the amount of available antibody that can bind to the full length. And so therefore what that's going to do is it's going to free up the full length to be able to bind to the host receptor, which is that TIM1, which I didn't ask you guys to memorize. Okay. Is that anything further with that? Okay. Grace?
Can we maybe go over what rig I is again?
Sure. So rig I binds to double-stranded RNA. That's the first thing. The second thing is it's an inducer of interferon beta. Okay, so it's an inducer of this antiviral response. So again, what rig I does is if double-stranded RNA is present, this helicase domain would bind to it and change the shape of this to basically unlock these card domains. Now that those card domains are exposed, those will dock to this protein MAVs, and we kind of said, like, there's events that happen here, right? We didn't really go into detail of what these events are. But basically, it's a bunch of protein-protein interactions, like we've seen throughout the semester. So when two proteins bind together, changes the conformation, leading to maybe an activation of one. So in this case, rig I binds to MAVs, that causes a change in the structure, which ultimately leads to docking and activation of, in this case, there's a couple of different things. One, NF-kappa B, but this is the side that we focused more on, activation of ERF3 and ERF7. And the ERF, again, is an interferon activation factor or transcription factor, essentially. And we said that these, when phosphorylated, that phosphorylation exposes a nuclear localization signal, allows it to cross the nuclear envelope, and now bind to promoters upstream of genes like interferon beta.
And how does Ebola interfere with this process?
So Ebola does a couple of things. Does anybody have ideas as to what Ebola actually does to block rig I? Amelia.
Binds the double-stranded DNA so it can't bind to rig I.
Good. So that's one. The nucleoprotein is going to bind to double-stranded RNA, during the replication process or transcription process, preventing that activation. What's the other event? Sabrina.
Does it bind to ERF3, IRF3, and it prevents the phosphorylation?
It does prevent the phosphorylation of it. It blocks the ability of TBK and IKK epsilon to activate it. That's true too. We didn't really talk about this mechanism so much other than to state this happens. There's one other event directly with rig I though. I'll go back to another figure, and maybe that will make more sense. Here we go. What's VP35 interacting with in this image? PACT. And so we see PACT come up twice,
Yeah.
actually, right? PACT seems to be a protein that will activate these double-stranded RNA binding proteins independently of double-stranded RNA even being present. It's almost like there's this redundancy or backup mechanism. So PACT interacts not only with RIG-I, but it also interacts with PKR to activate them independently of double-stranded RNA. VP35 binds to PACT, prevents it from binding to RIG-I, prevents it from binding to PKR.
Okay. I think that's what I was confused on. Just like the whole like pack thing.
Yeah, so PACT has, at least what this suggests is that PACT has two binding factors, and the ability of VP35 to bind to PACT prevents PACT from interacting with each of those binding partners.
Okay.
Sabrina.
So could you say that Ebola like blocks the interferon response or double-stranded RNA independent and dependent mechanism?
Absolutely, you could argue that. Absolutely. It's a really good point. Amelia.
So, just so I have this right, the PKR phosphorylates the EIF2 alpha or whatever,
Yeah.
and that blocks cap-dependent translation?
It does, and this is something that we had talked about. We've talked about this a couple of ways now, right? So here's an example of that. So this is showing. I'll turn off the lights. I don't know how much this will help since we got the blinds open today, but. So if you take a look at this example here, let's just look at with no VP35 and then with VP35 real quick. So with no VP35 in the presence of double-stranded RNA, here's total amounts of PKR, here is the phosphorylated form. So double-stranded RNA induces a phosphorylation of PKR, that allows for it to be active or on, and now it phosphorylates EIF2 alpha. That results in a reduction of the amount of GFP that's being produced. And GFP in this case is cap dependent. When VP35 is present, blocks phosphorylation of PKR, blocks phosphorylation of EIF2 alpha, allows for cap dependent translation to occur. And this would argue then too, Amelia, you were asking about, we didn't talk about translation of Ebola. This would argue that Ebola does undergo translation by a cap-dependent mechanism, and that by blocking this process, it allows for its mRNAs to be translated. Go ahead.
I have in that section of my notes something about PAP competitive binding VP35 for L.
Yeah, so this was, I realized I had kind of screwed up the notes here, actually. And Annie pointed this out. She had posted something up on Blackboard about it, and so I tried to address it. So let me make sure that I address it now. See, this reference here is for me to make sure I fix this the next time around. But what I found out is actually the two of them, PACT and VP35, are described as being mutually antagonistic. In other words, both of them inhibits the other one's function. So let me try to draw this out real quick. What we've talked about is that VP35 will bind to PACT, and that binding prevents PACT from binding to PKR, and it prevents PACT from binding to RIG-I. And we've talked about that as being beneficial for the viral replication, or for viral life cycle. But at the same time, PACT's binding to VP35 actually prevents VP35 from interacting with L. And what's the function of L? Good, it's the polymerase. And we know that this interaction between VP35 and L is essential for transcription and for replication to occur. So what's been argued in some articles is that actually the fate of an Ebola virus infection is in part based on the relative levels of VP35 impact. In other words, let's say within a cell there is just as much PACT as there is VP35. Let's say there's 100 molecules of each of them. All of them bind together. That blocks an antiviral response, but what else does it block? Exactly, it blocks the infection within that cell. What condition would allow for the virus to not only block an antiviral response, but allow for replication to occur? What would have to be true about the relative levels here? Yeah, more VP35, right? You'd want like a 10-to-1 ratio or something like that. So there have been some arguing that an effective immune response here is going to be dependent on the amount of VP35 that's being produced in the cell and how much PACT is present as well. Be really interesting to see in individuals who survive Ebola virus infections, how does PACT expression change in those individuals versus individuals who succumb to infection. $1 million idea for whoever wants to take it. It's probably really not $1 million, but does that answer your question, Amelia?
Yeah.
Okay. Sabrina.
I have in my notes that VP35 binding to PAC may regulate transition from replication to transcription.
Yeah, and that's I think the part that I have incorrect. I think that it's rather that binding of the two of them would reduce transcription and replication. So you could argue that that might be a point at which the virus is converting from biosynthesis into assembly. I don't know if that's true or not, and that's something that I would have to take a look at. Should I throw a question out there?
Sure. Go over the biosynthesis of HIV again, the jumping thing.
Yeah, I'll try to do my best.
Oh, yeah.
And while we do this, then, let's review the different functions of those different elements, too. So what's the function of PBS? Yeah, polymerase binding. Or you can also say primer binding too. What's the primer for the first part of the reverse transcription? tRNA, perfect. All right, so polymerases always move, or are always let's rephrase that. Always synthesizing genetic material in which direction? 5' to 3'. So they're moving on the template in 3 to 5. So in other words, with how this is primed and where the tRNA is binding to the PBS, the first movement here, go ahead.
Wait, you just said it 3 to 5 and then 5 to 3.
Yes, I did. It makes or polymerizes in the 5 to 3.
Yeah.
It reads in 3 to 5.
Gotcha. Okay,
Reads the
I was thinking
template. Sorry. Why can't you see what's going on in here, Jonah? So first movement is towards the U5 and the R region. Okay, and what do these end up making up? U5 and R and what else? Something else. They combine together to make up something. Perfect. So U5, U3, and R make up those long terminal repeats. So movements towards this end here. What happens to these DNA, RNA molecules? And so focus on this right here. What has happened? RNA is degraded how? What has the RNase activity? Yes, it is, exactly. The polymerase has what we call RNase H. And the H here is just, I don't know if I noted this really well before, but the H is just a subtype of an RNase. We classify RNases based on what they will actually cleave. So there are some that cleave single-stranded RNA. There are some that cleave double-stranded RNA, RNA-DNA hybrids. RNase H is one that does these hybrids. So we get degradation, and again, what's going to happen at this point is we're going to get basically a circularization event, where this R, now that there is R unbound, or complement of R unbound in the cDNA, this comes around, binds to it, creates kind of a loose circle, not closed, but a loose circle conformation, Sabrina.
Is it actually like nucleotide here?
Yeah, it does. And that's what's not being shown here. So this, we talked about this strong stop where there's this jumping event. What's really going on here is that we're getting a base pairing between this R and the complement DNA of the R. Okay? Some models do a much better job of showing this jumping than this one does. And then we get polymerization to the end. So that's our first part. At this point, obviously what we're going to have is the U5, R, and U3 created. Here's that. What happens to this? What happens to the RNA from which a copy has been made? Degraded. With the exception of which one? Why? It does serve as the primer. That's not necessarily the reason why it's not degraded. Again, the reason here is RNase H has a hard time degrading purine-rich regions, which this is. The PP stands for polypurine, so A's and G's. But because of that slow degradation, this now can serve as a primer for synthesis of the full-length double-stranded RNA, or double-stranded DNA. So from PP, we get polymerization. tRNA lyse at this point can be degraded because that's RNA, and we've got a DNA-RNA hybrid at this point. We get our second strong stop, or this, again, this partially closed circle conformation, kind of shown here. And then the end result is finish off polymerization in both directions to produce double-stranded DNA. And again, we've got long terminal repeats on each end. PBS, gag, pull on, other elements as well. Jonah.
So after the strong stops, do these, like the R ends disconnect from each other?
There's actually not a physical disconnection. That's how, I think that's how the original model was developed, is there was actually like a physical movement. Let me try to draw this a little bit to give you a mental picture if I can. Just want to make sure that I have. Okay. I'm going to put a prime here, just meaning that this is the cDNA. Okay? So here's what we have at the end of the initiation of that reverse transcription. The RNA polymerase from that tRNA primer has produced cDNA of U5 and R. At that point, the RNase H activity of the polymerase will help to degrade the RNA of the R and U5 regions. What's going to happen is that this end over here, which is shown hanging off right now, is going to loop around. Okay, so let me redraw this to help model that. So these physically base pair, Jonah, and now because we have a free 3' hydroxyl on the end of this cDNA, now this can continue to polymerize all the way around until it bumps into that tRNA. This gets degraded, and you can fill in the rest of the PBS with cDNA. And now you have the template for producing that second strand.
That makes so much sense now. Thank you.
You're welcome. Grace.
Does the virus use its own tRNA or does it use the host?
So it doesn't encode its own, right? So the tRNA comes from the host. But that host tRNA is actually packaged in the viral particle upon exit from the previous cell that it's infected. So it's already there loaded on to the mRNA, or to the RNA genome, ready to perform reverse transcription once the virus has entered into a new cell.
Okay.
Sabrina.
So that's because this is all partially encoded
Yeah.
in the cytoplasm.
Yeah. And again, the importance of the partial encoding here, there's a couple of things that are important. One, that protects the genome from degradation. It protects any sort of antiviral response as well. And this is a common feature among viruses that have RNA genomes, that their replication is performed in, we saw both with Ebola and with HIV that Ebola virus had its nucleoprotein coding the RNA, preventing recognition. Here it's within a capsid. We see other RNA viruses will actually perform the same sort of function, but will do it in like endocytic vesicles that are buds off of the endoplasmic reticulum, actually, as a way to prevent degradation of the RNA or to prevent the activation of an immune response. But the partial encoding means that you've got some protein; those capsid proteins have started to undergo a change that creates like pores almost. Why is that important here? What does that allow to come into the capsid or into the... Yeah, exactly. So that allows for the nucleotides to come in to catalyze this reaction to occur. Grace.
Can we go over the ribosome slippage?
Yeah. So the main probably the takeaway point from ribosome slippage is it's a way to encode for two proteins on a single mRNA. Again, eukaryotes only transcribe monocistronic mRNAs, and so the virus has to find ways to get around this. And ribosome slippage is one of those mechanisms. And we talked about here, we didn't go into this in great detail, but we said that two key features are a stem loop or a hairpin loop. This RNA is complementary to itself to allow for the formation of that stem loop. And then we've got this slippery sequence. In this case, it's a run of U's. And the idea, again, is that that slippery sequence, polymerases and ribosomes can, on a run of 3 or more consecutive nucleotides, sometimes miss one of them accidentally. So imagine this as, has anybody ice skated and gone directly into a wall while ice skating before? Right? So you slip on the ice, you go straight into the wall, and you bounce backwards a little bit. That's what the ribosome is essentially doing here. It's reading these 3 nucleotide codons at a time, bumps into the stem loop structure, and slips backwards a nucleotide on that slippery stretch, and allows for a frame shift, a -1 frame shift, meaning it's backing up a nucleotide, and now that's changed the codon read. And again, where this is occurring is within a linker sequence between the GAG and the Pol gene products. So Jonah had asked about, well, doesn't this have an effect on the protein? And yeah, it does have an effect on that linker sequence, but it doesn't have an effect on the fidelity or the amino acid sequence of the GAG products or the Pol products. That linker actually will get cut by the protease and basically just get degraded because it has no other function. And I think we kind of highlighted that. And go back a couple slides. Right here. So these last couple of products, this bottom one would be the example where some sort of ribosome frame shift has occurred, and where it's occurring is within this small little stretch of sequence that really has no important function. So we would call it a linker, amino acid linker, for example. Grace, is that?
It kind of does.
Okay.
I just have to look at it.
Okay. Sabrina?
What genes that you needed to make the same amount of this would work?
You're absolutely right. This wouldn't work if you were trying to make the same amount of the same protein. What mechanisms would work?
Like polypeptides?
Absolutely. Polyproteins ensure that every time an mRNA is translated, all the proteins within it are going to be made at equal amounts, and then using either a host or a viral polymerase to resolve that polyprotein into the individual mature proteins. Absolutely. Jonah.
Is each line on this thing here just like a different translation?
It's a different mRNA that would be, and each of those mRNAs would be translated. So what this is trying to show you is that you've got a number of them that undergo some form of alternative splicing. And we've talked about mechanisms that HIV uses to ensure that its genes are transcribed. One that can help to control expression as well would be alternative splicing. We also said that some of these larger partially spliced, which is this one here, or unspliced, are not exported from the nucleus on their own. And so what do they require to be exported? Rev and the Rev responsive element, which we find somewhere within the ONV gene. And so that allows for the full length and this ONV product to be exported. This also controls the timing of when certain genes are going to be expressed. This ensures that VPR and TAT and REV and NEF are all going to be expressed before those structural proteins are going to be expressed or translated. So Jonah, what we see with HIV is all mechanisms that contribute to protein expression, polyproteins, ribosome slippage, alternative splicing. All three of those have some sort of role in here. You could also include probably nuclear regulation of nuclear export as a fourth way if you wanted to as well. This by far is the most diverse of the mechanisms that we've seen, just with the variety that it's using.
Is this like characteristic of most viruses, or like
That it's this complex, or?
that it uses all like multiple mechanisms, or is it more?
I think that there's a range. So there are some like poliovirus, it only does, its mechanisms are pretty dang simple. It uses an iris for translation, and it produces a polyprotein. And then there are others like we saw with HPV, it does alternative splicing. And that helped to control the expression so that not all proteins were produced at the same time. And also, we talked about how the differentiation of the cells helps to control the early versus late genes. HIV is kind of on the other extreme in some ways. It uses ways to regulate which genes, uses ways to regulate how much of those genes are going to be produced. So, Sabrina, I think the short answer is there's a variety of complexity. Apparently that's the bell. Jonah.
GAG and Paul are polyproteins.
They are both polyproteins.
So they're broken apart by the protease, right?
Yep, which is on here, that is what's labeled as PR. So that's part of the polymerase gene product.
Is that protease included in the capsid originally?
It-
Otherwise, how would it?
It is, actually. So again, if we take a look at the capsid structure, remember that the GAG and GAG-PAL would all assemble still in an immature form. We get, in the model right now is that either exit and maturation are occurring at the same time, or maturation is occurring after exit. That's the time protease would be able to cleave that immature form into the active or mature form of the viral particle. And so that rearrangement, that cleavage process is actually taking place inside that virion.
So nobody asked questions about HIV.
You mean Ebola?
Mm-hmm.
Or HIV in general?
HIV.
No, there were some questions on
Oh,
HIV on the
no,
exam.
Guys.
It's about a 50-50 mix.
No.
And I might point out, do not forget to look at the first lecture from Ebola, where we talked about things like incidence, prevalence, epidemic, endemic, sporadic. Take a look at that stuff again, okay?
For like numbers for that, right?
No, I'm not asking you to know numbers. I'm asking you to be able to identify. So, like, you'll see the take-home has a question on incidence and prevalence and like prodromal stage and stuff like that, where you're actually going to have to analyze a graph. You could see questions come up on the test that might just ask you about definitions or descriptions of that terminology. So do take a look at that. Shania?
So, in like the beginning of the Ebola lecture, there was like that table, it was like the number of cases, and then like.
You do not need to know those numbers if that's what you're
how to
asking.
figure out those numbers, like, the percent kind of thing.
I think the key distinction to take away, I would give you the parameters, but remember the key distinctions are incidence represents the number of new cases, and prevalence represents the new and old. In order to understand prevalence, you have to understand how long is the course of an infection to be able to determine what range or window of incidences you could include to account for the prevalence. But if I were going to ask you, I'm not going to ask you guys to do any calculations where you have to do like per 100,000 or anything like that. I'm not going to worry about doing that. We didn't spend enough time with it, so. All right, I will see you all on Friday, BYOM as usual.